minutes to go through all that. I'm gonna do my best. So let's start by answering maybe simple, maybe even trivial question, but why is that weed identification actually so important? Well, it's actually a key to a successful control, not only with herbicides, but in general, successful control of any weed. Therefore, I think it should be a first step in our decision-making process, and if only it is possible, it should be done at an actual species level, because there may be certain differences between certain species within the same genus that will require us for taking various different approaches when managing such plants. Also, when we have the knowledge of an actual species, then we can also understand its biology, we can understand its growth and reproductive characteristics, and this will pretty much help us understand two more additional things. The first one is the actual reasons for why that weed is actually occurring in this particular situation. And knowing that, we can make some corrections, necessary adjustments to remove that original cause for the weed infestation. The second thing is that it tells us what are the actual strengths and weaknesses of that particular weed species. So just like the intelligence to the military, it will simply help us hit that weed where it hurts. And so, in other words, to choose the proper control methods. The point here is that the weed management techniques exploit or leverage the life cycles of weeds and use the biology of weeds and their characteristics in the development of hopefully efficacious control strategies. And so this is all why the weed identification is so important. Now, before we dive into the details, let's start here with answering the question, how can we easily differentiate grasses from other types of weeds growing in turf? So grasses are going to have the hollow, rounded stems, well, rounded or flattened depending on species. But definitely that thing that tells you it's a grass, it's that there's pretty much nothing within that stem. And also grasses will have very distinct joints, so nodes on the stems. Their leaves are going to be longer than they're wide. Broadleaf species, they're pretty much easy to recognize. There's a great variation within them, but usually what they have is net-like veins and showy flowers. Sedges and rushes, on the other hand, the stem in those plants are going to be solid. Sedges will have triangular shaped stems, so it's easy to recognize when you just try to roll these stems in your fingers. Rushes will have round stems, but again, those are going to be, there's going to be a tissue within them. So again, the point here, grasses will have those hollow and rounded or flattened stems. Among grassy species, we'll have mainly annual plants, which will complete their life cycles within one season, either from spring to fall. So those are going to be called summer annuals, or from fall to spring, and those we're going to call winter annuals. So the examples of summer annuals will include plants like large crabgrass, smooth crabgrass, goosegrass, barnyard grass, yellow, green foxtail, or sunbur. Winter annuals is going to be, for example, annual bluegrass. There's practically no biennial grasses, so the plants that are finishing their life cycle in 2 seasons. So in addition to annual plants, we've got quite a big group of perennial grasses. Well, all of our desired turf grasses are perennial. So those plants are completing their life cycle in 3 or more growing seasons. They're much more, they last much longer within a stand, and well, this trait is why we're using some of those grassy species as desired grasses. But the weed examples will include species like Dallis grass, thin or bull paspalum, nimblewheel, orchard grass, quackgrass, bahia grass, perennial ryegrass, and others. So as you can see, there's quite many examples that we're talking about. Obviously, we're not going to be able to spend enough time to go through all of them, but we're going to focus on some most problematic species as identified by Florida industry. So according to the survey that I have conducted a few years back in cooperation with Florida Golf Course Superintendents Association, we have identified goosegrass as the #1 weed for the Florida industry. 69% respondents indicated that they either have it or as the #1 weed or among top 3 weeds in their operations. Then we've got tropical signal grass as #2, torpedo grass, and crabgrasses. And crabgrasses and annual bluegrass, depending on where you're at in Florida, crabgrasses are much more common in southern part of the state. So that's why we're going to focus on crabgrasses, not annual bluegrass. The identification methods that we're going to discuss today are going to be pretty much the same for all the grasses, because we're going to be discussing the actual workflow on how we identify grasses, and then we're going to utilize those 4 species. So again, goosegrass, tropical signal grass, torpedo grass, and crabgrasses as our examples as we go through that particular workflow. So this brings us to diagnostic grass features. So we can talk about that in a little bit more detail. Now, looking at the grassy plant in general, grasses are going to be growing upright. They're going to be usually bunching plants with fibrous root systems. Some may or may not have rhizomes or stolons, and as mentioned, the grasses will have those hollow stems that are either round or flattened. The leaves are going to be arranged in groups of 2. They will have parallel veins. Sometimes this may get confusing because grassy plants are monocots, but they're not the only monocots. You will have some broadleaf species that are also monocots and can be easily confused with grasses, especially when there's no other identification species. And the one that I'm thinking about right now is doveweed. Doveweed is quite often confused with grasses just because those leaves are similar, and that venation is parallel in those leaves. Grassy stems are all, and I mentioned that already as well, are going to be almost always distinct because of the nodes and joints. Most grasses will have ligule where the leaf blade and the actual stem are going to meet. And grasses will have flowers organized either by spikelets, panicles, racemes. Now, speaking of those flowers, typically this is the easiest way to tell multiple grassy species apart. Just by looking at their inflorescence, seed heads. This term is used interchangeably. Depending on different times on the season, however, and the development stage of the actual plant during that season, or in presence of some external factors, those seed heads or flowers, inflorescence may or may not be present at that particular time when we need to identify that plant. So in turfgrass also, which is one of the management methods for turfgrass is mowing. So oftentimes those seed heads are just chopped off, and even if the time in the season is right, even if the growth stage is right, you will not see those seed heads just because they've been chopped off. And so oftentimes in turfgrass situations, we must rely on some other characteristics, some other traits of the plants to identify what actual species we're dealing with. So before we go into those other traits, Just wanted to show you some examples of those four species that we're going to use as our primary examples today. So goosegrass in your upper left corner, finger-like spikes growing mainly out of the same point, with one, sometimes two spikes growing out of the like below that cluster point. Those racemes are going to be a little bit wider than in other similar grassy species, and well, someone said that they resemble a goosefoot, hence the name of the plant, goosegrass. In crabgrasses, we will also have spikes, but they're going to be much thinner. We can have more of them, and the number is going to differ by the species and actual individual plant. For torpedo grass, this is going to be quite distinct example based, on the background of the other ones, because this is going to be the actual panicle. And tropical signal grass, this is also quite characteristic in fluorescence over here, because it's going to be a few branches, 2 to 7 branches along the straight stem. And those branches are going to be at 90 degrees to the actual stem, resembling a signal flag, also giving the name to that particular plant. But again, we may not see those in fluorescence, we may not see those seed heads in the actual field when needed. So in the absence of those, then we will usually start identifying our plants by what is called the vernation type. Not venation, but vernation type. And it's simply an arrangement of leaves in the bud leaf, so that newest emerging leaf and the surrounding sheath. In turfgrass, we have two types of vernation, either circular and rolled, or V-shaped and folded. And well, there are several techniques that we can use to determine that type of vernation. Usually the most reliable one is going to be by cutting, by getting either very sharp knife or scalpel and just cutting through across the stem and exposing that cross section. However, if we do not have a knife, we can always just simply peel back the leaf from that stem and see if the leaf sheath either curls into a circle or just simply falls flat. Another method is, well, this one is not as reliable, but we can simply try to roll that leaf bud within our fingers and see if it rolls easily, then the vernation is likely circular and rolled. But if it's not, then you can assume it's probably V-shaped or folded. However, you're going to have species like crabgrass, in which if you're not gonna roll the actual leaf bud, this may get confusing because they will have rolled vernation, but they may have also flattened stems. So not always reliable method, but when you look at a few of those plants and you're going to look at those leaf sheaths, peel them back, you're gonna figure out easily if that's a folded or rolled vernation. Why is it not moving forward? Okay, so just an example again. Our plants for today: crabgrass and tropical signal grass. Example of actual crabgrass emerging on the left-hand side, and goosegrass, torpedo grass on the right-hand side. So you can see that the new leaf emerging from that goosegrass plant on the right is definitely folded and V-shaped, while looking from the top at those plants on the left, you can see that those leaves are curling into that leaf bud, and so that is that rolled vernation of crabgrass. And tropical signal grass shared that same trait. So when you have V-shaped, you will either be looking at goosegrass or torpedo grass, circular, either crabgrass or tropical signal grass. So that's how you separate those plants into groups. The next thing we're going to be looking at is a ligule. ligule will be this protruding structure from the upper surface of the leaf, where the leaf blade and the sheath are joined. And if present, because it may be absent, but if present, that structure will be either membranous, or it will be very, very short membrane with a distinct fringe of hairs. There may be also some hairs around that ligule, which are not going to be the actual ligule. So if there are hairs present on any type of those distinct traits that we're going to be talking about, this is another identification feature, just the presence of hairs at that, in that vicinity. And those ligules may vary in both shape and size depending on species. So again, going back to our examples, goosegrass will have the toothed membranous with that very distinct and characteristic V-shaped cut in the middle. Crabgrasses and goosegrass that will be membranous ligule. Crabgrasses will also have membranous ligule, but there will be no cut in the middle, and it will be depending on the species, either longer or shorter. Torpedo grass, as you can see, there is no ligule there, and tropical signal grass will have the fringe of hairs. So like I said before, we've got that vernation. If it's folded or flat, then it's either goosegrass or torpedo grass. So when you try to distinguish those two species, if there is a membrane, toothed membrane, it's goosegrass. If there's no membrane, that's going to be torpedo grass likely, right? If we're separating between those species. Now for crabgrass and tropical signal grass, they will both have rolled vernation, and then you look at that ligule. If it's a membrane, it's likely crabgrass. If it's a fringe of hairs, then you're most likely looking at tropical signal grass. And if those are not helpful yet, then the next step is looking at the auricle, which is the pair of appendages protruding from the side of the grass leaf and the junction of the blade and the sheath. And auricles again may be absent, but if they are there, they may be rudimentary, so short and blunt, or there may be large and just clasping the stem and be like claw-like. Some may be again pubescent, have short hairs attached, and if those hairs are there, this is another identification feature. Leaf sheaths may be either open, split with overlapping margins, or closed. Now the leaf sheaths, as shown here, those are going to be much more difficult to correctly identify because the plant, depending on various external factors, may be looking different in that area. And so, for example, goosegrass in the upper left corner, you can see, well, this was supposed to be example of split but overlapping sheaths, but this particular plant did not have that overlap that was very distinct, right? So sheaths are going to be a little bit more confusing. Then the collar, some plants may have a very distinct collar, which is the space joining the leaf blade to the sheath, but usually, just like with the sheaths, it's one of those traits that may get a little bit more confusing. So, yep, leaf tips. Most of the examples we're talking about today are going to have sharp and pointed leaf tips. So tropical signal grass, crabgrasses, well, tropical torpedo grass mainly will have like very distinct sharp pointed leaf tip. Thinking about that, crabgrasses and tropical signal grass will have something in between sharp and blunt, depending on various factors. And then goosegrass will have very distinct blunt leaf tip. Of course, if it's not cut off by the mower blade, right? And then boat-shaped will be present in plants like St. Augustine grass, for example, or speaking of weeds, annual bluegrass will be probably the primary example of boat-shaped leaf tip. So those are, again, some traits that we can use to help ourselves if, while doing the identification, if we couldn't identify the plant by vernation, seed head, and the ligule, right? Then grasses can be characterized by both the self-supporting, upright growing, or prostrate, creeping, or trailing plants, right? And especially that second type of growth will be associated with the presence of either rhizomes or stolones. Now, both of these structures are going to be simply transformed stems. Rhizomes are going to be the ones that are growing underground. Stolones are going to be the ones that are creeping above ground, and they will both have the ability to create roots, and then give rise to the shoots at the nodes. Again, going back to our primary examples for today, goosegrass, as you can see, will have neither of those. Will not create rhizomes or stolones. It will create this circular rosette, wagon wheel-like rosette with whitish center, very very flashy leaf sheaths in the middle of that plant. Now, crabgrasses and tropical signal grasses are going to have the ability to creep, and they will create stolones, which are going to be, since those are above ground parts of the plants, they will be having that green tissue. Compared to torpedo grass, which creates rhizomes, so underground tissue, there's no light that they're being exposed to, so they're going to be white in color. All those three types of grasses, crabgrass, tropical signal grass, torpedo grass, they're very aggressive, and those structures are allowing them to sometimes perennialize and make them more difficult to control. So those are the most of the traits that are very important throughout the process. And here's the summary of that workflow that we've been talking about. So again, we start with vernation, identify if it's folded or rolled. Then we go and look at the ligule, if it's absent, fringe of hairs, membranous, if the hairs are present or absent. Then we move on to the auricles, absent, claw-like, hairs present or not, rudimentary, hairs present or not. And then we look at more deeply at stolones, rhizomes, if those structures are there. And then we move in to look into other traits like sheath, color, leaf blade, and hairs on leaves, maybe other identifying features that are very distinct to particular grassy species. And using that workflow, we can utilize multiple ID keys that are available online. This one here, it's coming from the NC State, and it was developed by Dr. Yelverton. Using the discussed workflow and such key, you can more easily narrow down trait by trait what is the individual species of interest of yours. So just using that top example from this slide, we have a plant, we're looking at the vernation, and it seems to be folded, right? So then we move on to the ligule. Ligule seems membranous, and there are some hairs over there. Okay, checked. So the next thing, we're looking at auricles, and well, there are no auricles, no stolones, no rhizomes. So we've got all those boxes checked. Then we look at the sheaths. Sheaths appear to be flattened and split with overlapping margins. The collar seems to be continuous with hairs on edges, and the leaf blade seems to be blunt. And all of those things, step by step, this will lead us to the conclusion that this plant is most likely goosegrass. As you can see, there is no mention of seed head over here. So those identification keys will allow you to narrow down the species of interest without looking at those seed heads. When you have problems identifying the plant, you sometimes send us photos to help you. So if you're going to be doing that in the future, sending photos that include these particular structures will help us help you and narrow down what is the actual species that we're looking at. Oftentimes, it's very difficult to identify either turfgrass or grassy weed species simply from just an image that, especially if it does not include all these structures. All right, so this would pretty much summarize our identification portion of this presentation. And just looking at the time real quick, I think we've got 20 more minutes. So we're going to try to go through the management options again for this, for those. Well, this time we're going to look at 3 examples: goosegrass, crabgrasses, and tropical signal grass, mainly just because torpedo grass is much more difficult to manage, and there's not that many options out there actually for selective control, at least. Just a few more general notes that I wanted to make here. I try to always make those points. Weeds are not a result. Sorry, weeds are not causing the turf to deteriorate. It's actually the opposite. It's the turf deterioration that allows weeds to encroach and to infest. So in other words, it's the lack of turf density or those areas devoid of growth or turf canopy that allow light to the soil surface and promote weed encroachment. So the point here is that unless these factors that are contributing to that turf decline are corrected, that continued weed encroachment should be expected. And when we think about that, flipping that all around gives us the primary method of weed prevention, which is simply by ensuring the proper growing conditions for turfgrass, right? So whatever we can do to optimize those growing conditions, figure out what is the optimal mowing height, irrigation, fertility, make sure that the turf is dense and of high quality. This is going to be the first line of defense against those weeds establishing in that turf. Mowing is another way of controlling weeds. We're not going to spend much time here. It's just that simply some weeds cannot withstand it, and then you're just gonna mow them out. And those weeds that we're talking about today, usually they can withstand mowing. So how we can incorporate mowing into the prevention program here is either by mowing the seed heads off before they form the actual seeds and start spreading, or when those seed heads are already out there, we can just utilize the basket. So our usual recommendation is for not using the basket while mowing, and we just return those clippings with nitrogen back to the plant. But if you have those seed heads, especially if you have plants that have very high capacity to produce viable seeds, then by just collecting those seed heads into the basket and taking them out of that yard will help preventing the further spread. Manual removal, whenever you can do it, it's always the best and most environmentally friendly method. It's important, though, to make sure that you are removing the entire plant. So not only the above ground part of it, but also the roots. And in terms of like plants that are spreading, it may be difficult because those rhizomes sometimes may create very complex structures that it's not easy to handle without destroying the yard. Another point I wanted to make before going more deeply into details about what are the chemical options is that the chemicals should be utilized as your last resort, right? So first, you're going to have layer of pre-emergence herbicides that are preventing preventing the encroachment or establishment of weeds, and then you're going to utilize post-emergence herbicides as the mean to control the ones that are already growing and are already emerged. But again, this should be while designing your management program, those should be the last points, like the cherry on top for the entire weed management program. And weed management should be incorporated into solid turf management program. All right, speaking of those pre-emergence options, like I said, a first line of chemical defense. This may be oversimplification here, but those herbicides have to pretty much create a preventive layer within the soil that that targets that it targets the radicle, so that first root coming out of the seed that is germinating, and this way preventing it from establishing an actual plant. And so with that being said, we have to remember that those options are going to be efficacious if we, well, one, apply them to the soil, so they need to be usually watered in and activated by irrigation. This is very, very important. Irrigation is needed for pre-emergence control, or rainfall at least, and they kill weeds as they germinate. So they are called pre-emergence, but they should be applied before germination because they prevent weeds from emerge. So when to apply them? Well, the example shown on the photo is the example of when it is too late, right? So if they kill weeds as they germinate, so once they germinate and once they emerge, even if they're not really much visible within the canopy, they're already out there, this is going to be too late. So you have to make sure that you make that first application before the germination starts, so before the expected window for germination, so that the plant is actually being killed as it germinates. So it is recommended to actually control or monitor soil temperatures and adjust the application timing based on the specific environmental requirements, and that is just because every year is different. However, in many situations, it's very difficult to do that. If turf management is not your first and only job, it's just going to be very, very challenging. So a rule of the thumb is, at least for us in Florida, that the first herbicide application should be February 1st in South Florida, February 15th in Central Florida, and March 1st in Northern Florida. According to literature, those timings usually coincide with the bluing of some of the landscape plants like azaleas or dogwoods. And for winter annuals, like Poa annua, schedule is going to be a little bit, not a little bit, but it's just going to be opposite. So we're going to apply those pre-emergence herbicides when the nighttime temperatures are going to drop to 55, 60 Fahrenheit. So early October in North Florida, mid-October in Central Florida, and early November in South Florida. So here you can see pretty much the explanation for why is that. So if the plant, like crabgrass, is germinating at pretty much 55 degrees Fahrenheit, then it starts germinating, and it's going to germinate throughout the time when the optimal conditions are provided by the environment, right? So you want to schedule that first application before that occurs, right? And pre-emergence applications will usually last some time, usually depending on temperatures and rainfall. The most common timing is going to be about 2 months. Some will last a little bit longer, some will last a little bit shorter, but usually about that time. So that one application, although its efficacy drops throughout the time, may still cover the entire, let's say, period of that emergence, right? But if you have multiple species, or if that emergence window is longer, then you will likely have to simply just extend that efficacy by reapplying your herbicide. And depending on different herbicides, it's always explained on the label. You may have different timings and or intervals to do that. By the way, those few slides are borrowed from my colleague, Dr. James McCarty at Mississippi State University. So just wanted to give him that credit over here. And if still have time, I think we still have 10 minutes. I would like to go through the actual options that you've got out there to control those most problematic species. We're going to start with the pre-emergence option summary, starting from crabgrass. And as you can see here, there's quite plenty of those herbicides, and we've got multiple mode of action groups that allow for different rotations. Most of the grasses that we are growing in Florida are going to be quite tolerant of those applications. So that gives us quite a lot of tools to control crabgrass. Again, you want to get those applications before the soil temperatures are going to reach 55 Fahrenheit for 4 to 5 consecutive days. Now, I just wanted to, we're not going to go through all of these just because we don't have time, but wanted to point your attention to oxidizone and indaziflam. Those are going to be a little bit hotter on some of the species. That's why you've got them marked with orange or even red color for some of them, meaning a little bit more caution is advised when using them, or there are some restrictions or some additional rules to be followed when using them, or simply a certain amount of phyto has to be accepted if using those options. Now, indaziflam and oxidizone are not going to be the preferential options for crabgrass, but those options, as you can see, before we're going to go there are also, this is just extension of that table, there are also some 2-way and 3-way premixes that you can utilize for crabgrass, so even more options. But going back to the topic of indaziflam and oxidizone, those are going to be more efficacious against goosegrass. So the situation is opposite here, right? And goosegrass will have a little bit less options that you can utilize. 2-way premixes in this case will utilize also some post-emergence herbicides that give you that ability to treat, especially with that second application, plants pre-emergence, and also kill off the plants that are already emerged. Goosegrass will germinate in 60 to 65 degrees Fahrenheit. So usually, this is just a generalization, but usually about 2 weeks later than the expected crabgrass germination window starts. So if you have crabgrass as a problematic species in addition to goosegrass, you're already covering goosegrass with your first application. If not, then you can probably wait a little bit longer before you put out those applications towards goosegrass. Tropical signal grass, even less options for the control of this species. Very important thing, its germination starts around 77 degrees Fahrenheit, so your options for the control of crabgrass and goosegrass will likely miss tropical signal grass. So you have to, if you have that species, either start a little bit later in the season, or extend your pre-emergence coverage to also include that species. Now, that species is a little bit more challenging because once it is established, it will also create the perennializing structures. Then, with pre-emergence options, you can control the seed bank or emergence from the seed bank. But if you have established perennial plant like that, pre-emergence options are not going to be able to handle that control, and you will have to utilize post-emergence applications. Speaking of which, let's go again and take a look at some crabgrass. Quite a long list of these options, but you know, there's a lot of red that you can see in this slide, which means either those are not not registered for that particular purpose, or simply are damaging to that particular grass. So although the list is long for the actual particular species, there's not too many options. Quinclorac, for some of the species like zoysia grass, quinclorac stands out as the go-to post-emergence application because it can also handle more mature and tillering plants. Another one will be pinoxidin, so manuscript for like Bermuda grass and zoysia grass. If you have San Agustin grass, unfortunately, at this time, your hands are tied. There are no true selective options that can handle established crabgrasses in San Agustin grass post-emergence. But yeah, this is pretty much the summary of options that you've got out there. And if there are going to be any questions, we can go back to this. Goosegrass, the most difficult weed to control in our situations. It's a warm season grassy species, so all of them are, but we're trying to get it out selectively from usually warm season grassy species. So not that many options for post-emergence selective control. Most of these will drastically lose their efficacy when the plant is allowed to mature and start to tiller. Goosegrass, although it's an annual plant, it will survive as so-called short-lived perennial in southern parts of the state. So pretty much remain green all year long, and it's just growing constantly, and you cannot really see the shifts with the populations, which plants were there before or not. So very difficult to control. So if you have the convenience of controlling it post-emergence, that is great. But if not, then post-emergence options needs to be applied, and as early as you start seeing those plants. If you're going to miss the critical window for application, then you just have your very risky phytotoxic options to the desired turf. So there's always some trade-off when controlling difficult species like goosegrass. One of the most standing out options is, well, Forum sulfuron is the current standard for zoysia grass and Bermuda grass. Then if this is going to fail, you've got metribuzin, and you've got also the option of using topramazone. Now, good news for those that are trying to handle goosegrass in San Agustin grass and zoysia grass. I want to point your attention to that section 2EE recommendation, trifloxysulfuron with safener only, plus fluazifop. This tank mix is actually Recognition plus Fusilade. Recognition is the new product that has a safener, allowing to use Fusilade safely in San Agustin grass and zoysia grass. And this allows you to remove Bermuda grass infestation from both San Agustin grass and zoysia, but also handles really well goosegrass infestation. So for the first time, practically, we've got some tool that can handle goosegrass infestation in San Agustin grass post-emergence. Going back to Bermuda grass, this is just an example of Sencor and Pilex and how well they handle goosegrass infestation as a tank mix. But again, with such a long vegetation in southern Florida, you may expect the reoccurrence at some point. And if you meet those caps on the label, you have to live with some of that infestation and handle it later on. Tropical signal grass, here the options are very limited. There's pretty much no selective options for the control of this species in warm season turf grasses. Well, there is amicarbazone, which is, quite theoretically at least quite safe to majority of these grasses. But then even the label is going to say that by itself, it's not going to be probably able to handle tropical signal grass infestation. You have to mix it with something like sulfentrazone, imidazole mix, and then it will decrease that, reduce that safety spectrum of turf grasses that you can use that tank mix on. And then we've got phenoxaprop that you can use in zoysia grasses. Then we've got pinoxidine that is very, very good product for the control of tropical signal grass, but works in in Bermuda grass and zoysia grass, and has some setting limitations. And then we've got sethoxidin, which you can use in centipede grass. In all these other grasses, you will be most likely left with the non-selective applications to the turf, to that weed, which will also injure your desired turf. So you will have to rely on spot applications of those products. Glufosinate works really, really good. And in some cases, like in Bermuda grass, it may allow also for recovery after a while. But yeah, non-selective options is going to be your go-to for tropical signal grass, and similar in most cases for torpedo grass, you're going to have to rely on those non-selective options, especially in things like San Agustin grass. So that's that. And not sure, I think I went a little bit over time. Oh, just one more example here, manuscript. So that pinoxidine used at what is called spot treatment rate. You can see how well it handled tropical signal grass in that situation. All around that plot is tropical signal grass. You can see Bermuda grass through, and there is quite clean Bermuda grass that is recovering those in those voids after TSG removal. It's pretty good treatment. So just want to leave you with some chemical options for homeowners. Not going to comment on that really, because, well, there's not much to comment on. And with that being said, I think I'm run out of time. So I'm going to do my best to answer any questions if there are any.
Wow, my brain is full.
Like I
The
said, 2 topics in 1 hour. So just did my best.
All right, let's see some questions here. Does the ligule serve any purpose? I guess any botanical purpose, any.
Well, um, good question. I'm not going to be lying. I don't have an answer to that particular question. It's probably more of a question to our herbologists. I'm not looking at those plants in that regard. I'm trying to figure out a way to identify and kill them. So from the botanical standpoint, I'm not going to be able to answer that question.
Thank you. Um, what is the difference between a key, a plant key, and workflow?
What is the difference between a key and a workflow? Well, the key is something that the key is the tool. Usually it's printed out, right? And you can use that key in multiple various ways. You not always have to use it as as our workflow presented, right? You probably may know already that your plant, it may be this or that, so you may start within the middle of that key, right? So that's how I at least understand the key as something that is published, and the workflow is just a way of looking at that key and going through it. I think those probably may be also used interchangeably, but that's how I tried to make the distinction.
Sure. Yes, this presentation is going to be recorded, so I'm going to send the recording to everybody so you can digest better the information.
Yeah. And
Great
and
class.
if not, then you know, you can reach out to me. I can send you portions of that presentation, especially I know most of people are interested in those summary tables. So that is something I can share. Also, I am translating those into actual ED's publications. Some of them are already submitted for review, so hopefully they will be coming out soon as well.
Yeah. Can you put your email address in the chat so we
Yes.
can have us looking information packet?
Okay. So
Can
I am putting my email address in the chat right now with the only note that this is the busiest time in the season for us, and we've got tons of research going on. So I'm not saying don't contact me. What I'm saying is just be patient. Sometimes it takes me a little bit time to respond. So I apologize if in advance if I cannot respond promptly. But I'm every month I'm going through all my communication, just trying to make sure that I haven't missed anything. Feel free if I don't respond right away. Just feel free to follow up. I'm gonna do my best to help every one of you.
yeah. If not, you can email me. I can maybe help. Another one here, and we're running out of time. Oops, what happened here?
I stopped sharing. Sorry.
Oh, okay. The chat.
So I can see the chat, and
Right.
the other question that I can see here. I'm not sure if I'm going in the right order, but is the azulog can the azulogs be used on zoysia grass? Well, officially, I cannot say it can because it's not registered. I myself have not sprayed azulogs on zoysia grass. But ultimately, some products, if they do not necessarily state that the product cannot be used on the particular turf, that means that may be labeled by omission. And as such, if any phytotoxicity or anything negative occurs, then the liability is on the applicator and the person who made the decision of using such product. There's quite a lot of products, so it's difficult to remember, the details about all of them. So, but I hope that answers the question.
Thank you. And this is the last one. We're running out of time. This is not the topic, but how you can ID Palmetto San Agustin grass from the other variety?
Um, this is a good one, and give me one. Well, so let me answer by saying this: the Dr. Kenworthy is our breeder at the University of Florida, and his crew actually created an ED's publication that is also submitted, and I've been included in that publication because I've been taking photos of detailed uh, plants parts to help you ID those species, not the species, but cultivars from one another. And I'm trying to find it real quick. Give me 1 second, really. Um, where is that going to be? Well, I've got the key almost in front of myself for that, but I'm not sure if I'm going to be able to find it in time before this presentation ends.
If not, we can do it later.
Okay,
We
sorry.
can.
I
I
mean,
have the email the people. We can do it
if that
later.
if that person is interested, in please email me and I can share that key with you to the identification of those.
All right. Let me try to launch the post test, guys. Take a look. The same questions. Hopefully, you guys can recognize the answer right now. Please take 2 minutes to find the answers. And at the end of the program, you're going to receive a survey. Please respond to the survey. This is the only way we know the feedback from you, so we can, schedule more programs, more in depth, webinars. So please complete the survey. So let's take 1 minute to finish this. CEUs we awarded the end. We have a pesticide CEUs 1 and FNGLA also 1. So I guess it's a very good packet for you guys. So let's wait.
And in the meantime, I found that slide. So
Oh, great. All right, let's wait 20 more seconds. You want to go to the answers, Pavel?
are we done with these, or there's
Give me
just
a
9 answers of 25? I can see the answers. Yeah.
Let's wait 10 more seconds. Right. Can you see it?
I can see them. Yep.
Right.
So the first question was, which of the following differentiates grasses from broadleaf, sedges, and rushes? And the correct answer was hollow, rounded stems. I've been trying to make that point 2 or 3 times throughout the presentation. That's one of the things we started the presentation with. Triangular shaped solid stems are of sedges, and net-like veins and showy flowers, those are going to be broadleaves. Which of the following is true for goosegrass? And the correct answer is that it has V-shaped folded vernation. I've been showing you the photos of the photo of the emerging plant that was on the right hand side and was like very flat. That leaf bud was very flat, and so that was that. Panicle was the torpedo grass seed head, and stolons were crabgrass and tropical signal grass. And then number 3, which of the following is true? Everyone answered correctly. So pre-emergence herbicides need to be applied before plant germinates. So no further comments here. The 4th one, which of the following is true? Goosegrass, crabgrass, or if tropical signal grass is germinating first before the other ones, and most everyone got it correctly. Crabgrass is the one germinating first when the soil temperature reach about 55 Fahrenheit, and then goosegrass at about 65, and tropical signal grass at 77 Fahrenheit. And I've been showing that chart from Mississippi State where it was kind of like visualized in time. But I'm happy that most of everyone got correct answers as the post. So hoping you guys enjoyed the presentation, and well, thank you so much, and hopefully see you next time.
Thank you very much. You are an encyclopedia.
Oh, no, far from that, but thank you. That's flattering.
All right, guys. Thank you a lot. Have a happy long weekend. Take care of yourself. Bye-bye.